Thank you very much. I am Antoni Sureda from the University of Balearic Island in Spain. And now I am going to present my work with the title The Invasive Lophocladia leyemandi Induces Oxidative Stress in Native Organisms in the Mediterranean Sea. Novel introductions of alien species have been accelerated in the recent decades by the rapid process of globalization and increasing trends of trade, travel, and transport. The Mediterranean Sea is one of the most affected areas by introduced species in the world, mainly due to its strategic situation and its semi-enclosed sea characteristics. In fact, invasive seaweed is a major global concern since they are known to deeply modify marine ecosystems, having a strong ecological and economic impacts. The slide shows some of the more representative invasive algae in the Balearic Islands, such as Caldera patacifolia, Alimeda crasata, Asparagopsis armata, and Lophocladia leyemandi. In fact, and among them, the red algae Lophocladia leyemanda stands out. This alga is considered as an alien species in the Mediterranean Sea and probably was introduced via Suez Channel and is widespread through the tropics and subtropics. Lophocladia leyemandi is a very aggressive invasive species growing as an epiphyte on other algae, phanerogams, and sedentary animals. In addition to altering benthic communities, reducing light availability, or hamper water movement, these algae produce lophocladins, alkaloids with important cytotoxic effects. Altogether, these algae can induce a situation of oxidative stress in the species affected. The aim of our study was to evaluate the effects of these invasive algae on native species of the Balearic Sea, such as the endemic seagrass Posidonia oceanica, the sea urchin Paracentrotus lividus, and the endemic fan mussel Pinna nobilis, using biomarkers of oxidative stress. First of all, I'm going to present the results obtained on Posidonia oceanica. This image is a representative sample of Posidonia oceanica epiphyted by the invasive Lophocladia leyemandi. It is observed that in the presence of the invasive algae, there is a significant decrease in the mean shoot densities, which is indicative of the mortality of the leaves associated with the epiphyte. In addition, it is observed that all oxidative stress biomarkers, catalase, superoxide dismutase, glutathione peroxidase, and the levels of glutathione are elevated in the presence of the invading algae. In addition, the production of hydrogen peroxide, as well as the oxidative stress markers, such as malondialdate as indicative of lipid peroxidation, and protein carbonyls as indicative of oxidation or proteins, are also elevated in the presence of the invasive species. In the case of the sea urchin Paracentrotus lividus, there is a decrease in the number of individuals in the area with the invasive algae compared to a clean area without the algae. Even the density is lower than in contaminated or polluted areas, showing that sea urchins avoid the presence of this algae. Malondialdate follow a contrary pattern with higher levels in areas with the invasive Lophocladia. Furthermore, the activity of the detoxifying enzyme glutathione S transferase is also higher in presence of the algae, indicating a defense mechanism against the production of lophocladins. As with the Posidonia oceanica, a significant increase in all antioxidant enzymes were observed in the presence of the invasive algae. In this slide, you can see an increase in catalase, superoxide dismutase, glutathione peroxidase, and also glutathione recrutase. Finally, the third species we studied is the fan mussel, Pinna nobilis. This is the largest endemic bivalve in the Mediterranean Sea. And we observed that Lophocladia leyemandi is capable of growing on the fan mussel, causing a significant physiological stress in the animal. The slide shows an image of the bivalve, of this bivalve in normal condition on the left side and covered with the invader on the right. Similarly to the previous results, all the biomarkers analyzed, including the activities of catalase, superoxide dismutase, and glutathione transferase, as well as the levels of malondialdate, were significantly higher in the gills of Lophocladia, in the gills of Pinna nobilis affected by the invasive Lophocladia leyemandi. And to conclude, we can say that the presence of the invasive algae Lophocladia leyemandi induces oxidative stress in the affected native species, which could pose a serious problem for diversity. Future monitoring studies on the long-term effects of the presence of the algae and development of polycyte to reduce its dispenser are necessary. And thank you very much for your attention.